Hello, my name is David, and today we will be doing an experiment with milk. For today's experiment, we'll be using three dishes prepared with agar, two milk samples, one of which was left outside in the summer heat for about a day, and the other was left in the fridge. I've made sure to use the same milk samples produced on the same day from the same place to ensure that all control variables were the same. Additionally, we will need time to allow our cultures to grow in the petri dishes. As mentioned before, with the two types of milk, we will be using experimental variables. I want you to ask yourself if you really know what an experimental variable is. An experimental variable is something that changes from experiment to experiment. It's essentially the thing that we want to test the effect that it has. So today, our experimental variable will be the milk. I would like to see how much bacteria is in milk after it has been outside for a certain amount of time. We will be using regular milk to show the amount of bacteria that is in the milk in the fridge when it's safe to consume. Additionally, we have our control variable, which will be our first petri dish. We use a control variable here because I have prepared these petri dishes myself. I hand washed, boiled, and put them in the oven to try to keep them as clean as possible, though it's likely that it's not perfect since I did it at my own home without much prior knowledge. It was my first time, so I still have a lot to learn, and of course practice makes perfect as shown here. The last part of our experiment today is going to be contamination. Contamination is a real part of any lab, not only contamination, but error. We are only human, and therefore we are bound to make mistakes about everything we do. It's for that reason that we should acknowledge the fact that we likely will make mistakes in our lab, and we should be on the ready to try to course correct. There are many opportunities to make an error or do something stupid or silly, so it's important that we not only keep these in mind and be aware of them, but also know that it is our responsibility to try to do our best to bounce back. Every lab will likely have some kind of error that keeps us from getting a perfect result. It is not expected that we get perfect results, especially as students, but we should just strive to follow our procedure and try to get our experiments as accurate and precise as possible in every lab that we do. But regardless of the contamination in this lab, you will see why it's absolutely necessary to put your milk away after using it. In the sample alone, you will see the amount of bacterial growth, how clumpy it is already after just spending a few hours in the sun. Over time, the bacteria will grow, even in the fridge. That's why milk spoils over time, even if it's left in the fridge. At the end of this lab, you will see why it's completely and utterly necessary to put your milk away. Bacterial growth is a very common thing in milk, and it's the reason for its iconic sour and clumpy texture. The bacteria that grows in milk that feeds off the lactose, the same sugar that many humans are incapable of digesting and that causes lactose intolerance, leads to lactic acid production. Like many acids, such as lemon juice or vinegar, it has a very sour smell and taste, while basic solutions like soap have a more bitter taste. It's for this reason that the quality of the milk is often linked to the way it smells and the way it looks, because bacteria often produces odors as it digests and breaks down its food. The aforementioned acid, lactic acid, is the same substance produced by our cells during strenuous activities such as exercising. That's what leaves your muscles feeling tired and fatigued. It's a result of your cells not getting enough oxygen and having to switch to energy production that does not require oxygen. That's why we pant so much when we exercise. It's so that our cells get the needed oxygen to keep on producing ATP, the energy required to function. It is my hope that at the end of this lab you will see the necessary components to every lab: precision, keeping an eye out for possible errors or contamination, following the procedure, and always being careful. I had experiences in labs where I didn't fully understand how to use the equipment properly, and I turned a knob too quickly, and the whole thing caught on fire. It's a Labs are very interesting and they can be fun, but it's important that you use and exercise the proper and very much needed amount of caution to when handling anything. Not only to ensure that your lab goes well and you can produce a high percent yield, but also to ensure your own safety. Because as often as we think of school as a safe environment and how we think of labs as being in our control, things can quickly run away. So it's important to always know where your hands are and to always know what is going on in front of you and around you. It's for that reason, when I pour my sample into the petri dishes, I will hold my breath to try to keep as much contamination out as possible. Additionally, I've washed my hands as thoroughly as possible. Unfortunately, I don't have gloves, but I've done my best with what I have. So let's get pouring. So I will pick a little bit of milk up, put it in there. I would like to say that I will not add a lot of milk because I do not want the possibility of mold growing from the moisture. After each use, I will mark each dish appropriately. On the first dish, I will add nothing, as I said earlier. This will be our first control dish. That way we can see what kind of bacteria will grow due to lack of sanitation, lack of proper and further sanitation that I was unable to perform at home. The next dish will have the safe to drink, perfectly regular milk taken right from the fridge. This will show how much bacteria is in regular consumable milk that is safe to drink. And this one will have the milk with high amounts of bacteria already grown in it, as seen by the clumps formed in it already. This one, I will remind you, was left outside all day in the hot summer heat. And I will remind you again as well that I will pour quickly and keep these lids on as much as possible to minimize the risk of contaminating it with bacteria from the surroundings. Here we go. I have a little bit of this here, and I'll pour it as quickly as I can. Okay, there we go. I will label it 1. Or 3, I apologize, 3. 3 tally marks, just like that. It's very recognizable, and I will remember. Now on the next one, just the regular milk. I'll take great effort to do it quickly. Now we'll label this one too. And in the last one, we will get no milk so that we can see if anything will grow naturally due to, as I've said, lack of sanitation. A few days have passed since we poured the milk into the petri dishes, and now we can look at our results. Some important questions to ask yourself, or for me to ask myself, is did I give this sample enough time to grow? These samples were left here in my kitchen for a few days, although maybe they could have used some more time. Additionally, did I provide the right substrate for them to grow in? Although many cultures of bacteria are grown in agar, maybe I should have seen how I can incorporate lactose in some way, since that's the sugar they naturally feed off of in milk. Additionally, I could practice pouring my agar, I could practice preparing my petri dishes, and work more on sanitation, although infestation and contamination wasn't really a problem here since there wasn't a whole lot of time for everything to grow. At the end of every lab, it is absolutely essential that you ask yourself, what can I improve? What didn't go right? And how can I do better next time? Even when you did things right, try to find some kind of takeaway that you can carry on to your next lab to ensure that it goes better. Even if it went well, you can always do better. Chemistry is all about reactions and procedures in lab. How will you react when your reactions didn't follow your procedure? Thank you. for watching. I hope throughout this lab I've left you with some meaningful insight on what it means to perform a good experiment. Although our experiment didn't go exactly as planned, I hope I still left you with some things to chew on and think on. Additionally, I hope I've shown you why it's important to put your milk away. This milk has been stinking up my apartment for about 3 days now, because I didn't know if I wanted to throw it away or not. So I'm eager to throw this out. Thank you for watching, and I hope that you put your milk away. Otherwise, you might have some kind of culture of bacteria growing, and it's not the good kind. Thank you for watching.